Hello everybody, here's a little bit of feedback to the ICT test for the Enzymes and Metabolism module from week 35, and I will go through the questions. So you have the question and the correct answer is highlighted in green. So here the first question is, which one of the following statements is correct? And of course the statement is that enzymes can have substrate specificity and or substrate or reaction specificity. So the enzyme can either can catalyze a specific substrate. That is not what I wanted. So we can have a substrate, so the enzyme can work on, say, a DNA or on a small molecule, like, for example, pyruvate. So they are very specific for the substrate, and they can also catalyze specific reactions. So, for example, a hexokinase would transfer phosphate residues onto basically 6-ring sugars. And for some enzymes, it doesn't matter whether it is glucose or galactose or something like that, as long as it is a 6-ring and as long as the substrate can bind to the active site. That's why this question would be correct. Okay, what would happen if the total concentration of enzyme was doubled in a reaction? Well, we said that the rate Vmax, for example, it depends on the rate, the K cut, times the total enzyme concentration. And therefore, if you increase the total enzyme concentration, let's say by a factor 2, then we would also increase the Vmax. If we decrease the total enzyme concentration, say by a third, then Vmax would decrease by a third. If we look at, for example, Km, however, Km is just simply the combination of three rate constants. So it is the combination of the forward reaction, so K1 plus K3 divided by K minus 1. And there is no enzyme concentration given in this Km. So that means that Km does not change. The only thing that changes is Vmax, and it goes in the same direction as the total enzyme concentration. So if we double the enzyme concentration, Vmax would double. And that's very important to understand. So what does Vmax actually tell us? And that is in the next question. And it tells us how the enzyme behaves at very high substrate concentration. So we've got our Michaelis-Menten equation here, and we say the substrate concentration is very, very high. Therefore, we can write Km plus S. This is approximately the same as S, because S is so much bigger than Km. And then we can write our Michaelis-Menten equation and can write Vmax times S. And instead of Km plus S, we just simply write S. So we ignore this Km because it's so much smaller than S, and we can cancel out S. So in this case, Vmax tells us how the enzyme behaves at very, very high substrate concentration. In fact, infinite substrate concentrations, but we would never reach these infinite substrate concentrations. This question asks about feedback inhibitors, and I showed you this diagram in the lecture where we have a typical case of feedback inhibition where compound B is generated from compound A in a reaction, and then B will inhibit the production of A. So what happens, and I showed you this diagram as well, what happens when we plot the concentrations of A and B over time is that at the beginning we see an increase of A, and then A will go down because the production of A is inhibited by B. And B will go up, but it will also go down because B sort of will inhibit its own production. And sooner or later, what we will find is that these concentrations will be quite stable. So negative feedback or feedback inhibition leads, in fact, to homeostasis. Homeostasis means that there are no changes in the concentrations. And it is an important mechanism to control how fast things run through a metabolic pathway. This question asks about the binding of an end product to an enzyme's allosteric site, and we remember that, so here we've got, for example, an active site, and we might have an allosteric site where we can bind an allosteric effector. And now when this happens, when this binding occurs, actually, what we might see is that this bound effector initiates a conformational change in the enzyme. It could, for example, close the active site, or it can make the active site more accessible for the substrate. So the important thing is that any binding of anything to an enzyme could cause a conformational change, and that then, of course, might affect the catalytic efficiency of the enzyme. This question asks you about an Eadie-Hofstee plot, and if you remember correctly, then in an Eadie-Hofstee plot, we plot the rate over substrate concentration versus the rate, and what we find in an Eadie-Hofstee plot is more or less a straight line. Now we said that the extension of this, the y-intercept here, would give us Vmax. That is Vmax, and we said Vmax is basically when the substrate concentration is very, very high, as we explored in the previous question. In fact, this Vmax we would only get if the substrate concentration was infinite. This point here in an Eadie-Hofstee plot is Vmax over Km, which tells us how the enzyme behaves at very low substrate concentration, and the gradient here, that would be negative Km, and Km tells us about the affinity of the enzyme for the substrate. An uncompetitive inhibitor, it will always decrease Km, because for an uncompetitive inhibitor we can write the mechanism as enzyme substrate, and that gives us enzyme plus product, and the uncompetitive inhibitor binds exclusively to the enzyme substrate complex. We have an enzyme substrate inhibitor complex. Now if we remove this enzyme substrate here by binding to the uncompetitive inhibitor, we basically shift the equilibrium, we move this equilibrium to that side, and that manifests itself in a decrease of Km. It apparently looks like the enzyme has a higher affinity Decreased Km means a higher affinity for the substrate. And in a way, this makes sense, because if we remove this enzyme substrate, then we will shift the equilibrium to that side, and it looks like that the enzyme likes to bind more substrate, which of course is true, but this here is a dead end, and therefore we will not produce a lot of product. So that's why we decrease Km. If it was an uncompetitive inhibitor, then Km would be increased, or the affinity would be lowered. In this question, we ask for the components of NADP, so nicotinamide dinucleotide phosphate. So we've got a phosphate here. We've got the adenine group here. So ribose and adenine. We've got two phosphates, and we've got a nicotinamide group here. So we have nicotinamide and adenine. So A and C here would be correct. There's no coenzyme A or riboflavin in it. Where in the eukaryotic cell would you find glycolysis? Well, of course, glycolysis is happening in the cytosol, in basically every single cell in the human body, including red blood cells. There is only in the cytosol where this happens. And this question here asks about what are the net products of aerobic glycolysis. And aerobic glycolysis means that we do not convert the pyruvate that we get at the end into lactate. So what we get really here is, when we look at the reaction, is we need to invest 2 molecules of ATP in the glycolysis, but we get these 2 ATPs out again because we run the glycolysis twice this process. So we invest 2 ATP, but we get 4 ATP out. We get 2 molecules of pyruvate, because pyruvate is a C3 body, from 1 molecule of glucose. And we also get 2 molecules of NADH, which we have here in this step. So we get 2 molecules of NADH, which we then can funnel into the respiratory chain. So this would be here the correct answer. We don't get the lactate because we are under aerobic conditions, and we don't need any fermentation processes that convert the pyruvate into lactate. In this question, we are asking about the function of the enzyme glycogen phosphorylase, and what happens with glycogen phosphorylase, it uses actually a phosphate residue to break the bond, this alpha-1,4 bond between 2 glucose molecules in glycogen, and it transfers the phosphate group to the 1 position of this glucose molecule. And the idea is, if you used water to do the hydrolysis, then we would lose the energy that is stored in this bond here. But since we use phosphate and carry out a phosphorylasis, not a hydrolysis, a phosphorylasis, we maintain this energy, and we've got the energy now stored from this glycogen, from this glycosidic bond. We have stored this energy in this 1 phosphate bond here, which we then, of course, can transfer to 6-phosphate, and we can prevent actually the glucose from leaving the cell. So it's a very clever trick. This here was a rather tricky question. We find that delta G under physiological conditions is negative. So what does negative delta G mean? So if delta G is negative, it means the reaction can happen spontaneously. Reaction spontaneously. So the reaction will actually happen. Now, if we've got a reaction A plus B, and we have the potential to run the reaction in both directions, we find that under standard conditions, delta G node prime is, we say it is positive. So it's larger than 0. So under standard conditions, this reaction would be on the left-hand side. So how do we get this reaction so that it goes to this side? Now, the only way we can do that, and we know that it is on that side, because we know that delta G is negative, and therefore the reaction is on that side. The only way we can do that is that we remove one or both products from the reaction, because then, according to the principle of Le Chatelier and Brown, we will shift the equilibrium to the right-hand side. So therefore, the way how it works basically in metabolism is that the products of the reaction are fairly quickly removed. And then it doesn't matter that delta G node prime is positive, and under standard conditions, the reaction would be on the left-hand side. If we remove the products, we force the reaction to go to the right-hand side. So what is the product of glycolysis in red blood cells, which do not contain mitochondria? And here this would be, again, glycolysis. But in this case, in the glycolysis in red blood cells, we are no longer under aerobic conditions. We do not produce the pyruvate. Pyruvate, in this case, as we discussed in the practical, plus NADH, H plus, is converted to lactate, plus NAD plus. And this lactate then is transported to the liver in the co-recycle. We need to carry out this reaction because we need to recycle the NAD+. And that is really the only way in red blood cells to recycle NAD+, which we know we need for the glycolysis. If we don't have NAD+, we can't run the glycolysis. If we can't run glycolysis, we cannot produce ATP. So in red blood cells, or cells that in general cannot do oxidative phosphorylation and get the NAD+ through oxidative phosphorylation, we need to recycle it and gain this NAD+ that it can be used in the next glycolysis round. Where in the cell would you find the enzymes for beta oxidation of short chain fatty acids or medium chain fatty acids? This is in the mitochondrial matrix. There is a slight exception. Very, very long chain fatty acids are usually broken down in specialized organelles, the peroxisomes. That's why I said here short chain fatty acids. But here the enzymes are all found in the mitochondrial matrix. We discussed, for example, to transport fatty acids into the mitochondrial matrix, we need the carnitine transporter. This question is which compound is not an intermediate of the citric acid or the Krebs cycle? And I told you the mnemonic, can I kiss Susie's sexy friend more often? So it basically contains all the compounds of the citric acid of the TCA cycle. What you don't have in it is lactate. And lactate is not a compound of the TCA cycle, nor is pyruvate. Pyruvate that we have here is converted into acetyl-CoA. So even pyruvate is not a substrate for the TCA cycle. And the lactate, that would basically be the step before the pyruvate. So we have oxaloacetate, we have citrate, we have succinyl-CoA and alpha-ketoglutarate, all in the TCA cycle, but not lactate. So I hope this makes sense and it helps you to understand the questions from this little test. Thank you very much for watching.